Hello, everyone. Today we're going to be discussing something known as the Hill reaction. As you can tell from the subtitle thing below, it's an exploration. Well, yeah, what we're going to be doing is an exploration of important experiments in photosynthesis. So Hill reaction is one of those experiments that have been done, and it's really important, really influential, and it helped to develop a lot of the ideas at the time. As you can see, we have an I here, and this is not the letter I, but it's a Roman numeral for number one, and this alludes to the existence of forthcoming additions to the series. So yeah, what you're watching right now is the first in a three-part series. I have three experiments planned to explain, and this is the first one. So be sure to check out the next. And after we go through each experiment, it's going to be much more difficult and much more complicated. So this is the easiest one to learn and understand about. Yeah, so we're gonna just start with some history first. Um, as you can see, the experiment, it was named after the scientist who discovered it himself. His name is Robin Hill, a British biochemist, born in 1899. And yeah, just some historical context towards the experiment. It was done in 1937, uh, and at that time, people they didn't know a lot about photosynthesis yet. Not a lot of research has been done. Lots of limited knowledge. Basically, I've summarized the five things that were known to them at the time. Number one, photosynthesis happens in the chloroplast of the palisade mesophyll cells of green plants. Number two, it requires light. Yep. They know that it consumes water and carbon dioxide, and it produces carbohydrates and oxygen. So basically, the formula that you're all supposed to learn in science class, grade 4, grade 5, whenever that is. And next, it is a redox reaction. So you can tell, I mean, if you've taken a chemistry class before, it's pretty easy to tell that water is being oxidized and carbon dioxide is being reduced. So there is that transfer of electrons, and that's something that they figured out as well. And that's basically it, just 5 sentences. So if you happen to travel back in time and ask the most prolific, smartest scientists around at the time to summarize the entire process of photosynthesis, they can 100% with confidence answer with these 5 sentences, and that because that's all they really know, that's all they could really prove at the time. Funny thing, 100 years later, no, actually, that's 80 years later. Yeah, 80 years later, you could ask a middle school student the same thing, and they'll most probably give you the same answer. So, yeah, things change pretty quickly, especially in science and research. A lot can happen in 80 years. And this experiment is really important because it really expanded the knowledge of photosynthesis beyond just these 5 sentences. It answered some of the mysteries that were questioning scientists at the time. There was huge debate, huge controversy between whether the oxygen that came from photosynthesis, right over here, whether it came from carbon dioxide or whether it came from water. So you see there's two possibilities. We have two water molecules here. The oxygens just happen to combine and it releases hydrogen ions. That's one possible mechanism. Another way is if the carbon were to break off from the oxygen, and the oxygen on either side just combines with each other and forms O2 again. And they really didn't know which reaction was proceeding, the first possibility or the second possibility. They have no way of finding that out too. Next, they were also questioning whether photosynthesis happened with separate processes, or if it's just like one single continuous process. So a lot of people, they say, oh well, maybe carbon dioxide that goes through a single process in the cell, and water that goes through a separate process. So like these things here, yeah, they represent different processes. So after they're individually processed, then some stuff happens and you get the products of photosynthesis. Next possibility is if both of these undergo changes through the same cellular process, and that results in the products of photosynthesis. So of course now we know the answers to that. We know that it's water that creates the oxygen, and carbon dioxide has no role to do with it. And we also know that the process is actually separate. We have light-dependent reaction and light-independent reaction. So the Hill reaction, yeah, the experiment that we're discussing, it allows us to prove that separate processes are happening, and it also allows us to prove that water is the source of the oxygen. So this thing here, yeah, this is what's actually happening. Alright, let's move on. So what's the experiment? What did he do? Right, so he took a bunch of leaves, right? And he had a way of separating, he had a way of not separating, extracting would be a better word. He had a way of extracting chloroplast from the cells. So he wanted to, experiment with them. He had these chloroplasts extracted with a method. We won't bother with the details, but we will just have a little review of the structure of the chloroplast before we go on. You're going to see there's many labels here, but we're going to just concern ourselves with two of the labels, the stroma and the granum. So inside the chloroplast, you're going to have this liquid, and that's known as the stroma. And this thing is surrounded by a membrane, right? So the granum is basically a smaller capsule within this membrane-bound organelle. So you have these tiny little capsules that are also made up of membranes. So it's just like a membrane within a membrane, and they go up in stacks. Each individual one is called a thylakoid, and when they are stacked together like this, that's called a granum. So this is also one of the mysteries at the time. How does the chloroplast work? What exactly does the stroma do? What exactly does the granum do? They have no idea at all. So what Robin did was he broke the outer membrane of the chloroplast that he had extracted. Yeah, so he extracted the chloroplast. Next, what he did was he broke the outer membrane. That way, all the stroma, all the liquid that's held within this membrane, it just spilled out, and you filter it, and yeah, all the liquid gets filtered out, and all you're left with is the granum. So yeah, he got just the granum. And what did he do next, right? He wanted to experiment on the granum. He wanted to see, what does the granum do? He wanted to answer that question. So he knows that photosynthesis, it requires carbon dioxide, it requires water. So that's what he did. The granum that he extracted, he added carbon dioxide to it, he added water to it. The carbon dioxide, I think it was like bubbled through the solution. And he waited, and nothing happened. No oxygen was produced. He couldn't detect any sugar. And at most points people would just stop here. They'll just give up, and they'll just write the granum is the useless part of the plant. It doesn't do anything. No, but no, he's a smart guy. He decided to go further with this exploration. So he's thinking, right? Uh, Maybe the granum doesn't work because I took the stroma away. Maybe the stroma has something that the granum needs in order to actually do work. So what he did was he added iron to it, an iron 3 plus solution. So it can be any iron salt. I think he used iron oxalate, but he also tried it with different irons, different anions, different salts, and he got the same results basically. So what happens was, after he put the iron, he got oxygen but no sugar. So the reaction happened in one part but not in both parts because the carbohydrate wasn't being produced. So why did he add the iron? Why did he choose to add the iron? Well, basically, he knew that photosynthesis was a redox reaction, so it has something to do with the transfer of electrons from one molecule to another, transfer of charge, that kind of stuff. So, like I said, something in the stroma is missing because, well, we just have the granum here. So maybe the stroma has something to do with the movement of the electron. That was like his hypothesis or something. So he compensated for that by adding the iron, because the iron, it can get oxidized, it can get reduced, and in this case, the iron 3 plus that turned into a 2 plus after this reaction was completed. And after the iron has allowed the movement of electrons, because that couldn't happen before. There was nothing to accept the electron with. We can generate oxygen now, but you still got no sugar. So he said, right, there's no sugar, all right? So what exactly does this carbon dioxide do? Because carbon dioxide is supposed to be our source of carbon, but our source of carbon is not being used to make the sugar. Sugar is made up of carbon. So if no sugar is produced, that means carbon dioxide, that really has nothing to do with the equation. So yeah, he removed carbon dioxide, he no longer bubbled it through the solution, and all of these things, it works just fine. So immediately you can tell that oxygen, that came as a result from water instead of from carbon dioxide. And yeah, he experimentally proved that. And through this, he also experimentally proved that two separate reactions were happening. This whole process here in the granum, the granum did something to the water to make it turn into oxygen, to separate the water molecules and turn it into oxygen. And that's the job that the granum did. So next he had a paper published, and this is just an image that I directly took out from it. And so he predicted that there were two reactions that were happening, and he thought up of the molecule A. He thought that there needed to exist a transport molecule that basically, after the water turns into oxygen, after the water is split, I have the reaction here. So you see the water is split into oxygen. You also have hydrogen ions and electrons. That hydrogen ion and electron, that gets accepted to a transport molecule A. Now we know that the transport molecule indeed does exist. Its name is NADPH. Yeah, NADPH2. That's the reduced form, and the oxidized form is NADPH. So he hypothesized the existence of the wait no. So he predicted the existence of the NADPH2 molecule before it was discovered. So he said that the transport molecule is needed, and that way it can move over to different parts of the chloroplast in order to reduce its target molecule B. So in the end, B gets reduced and it oxidizes A. So A is back into its regular form again, and A can just cycle back towards here for the first reaction again, and the same process can happen. So it's just like a transport chain of hydrogen that allows the reduction to happen. So presumably B is carbon dioxide, and carbon dioxide gets reduced to form the sugars, but why does that not take place here? Well, firstly, it's because you don't have the NADPH, and that's because this is just the granum. The NADPH that exists in the stroma over here. So you have NADPH molecules. They pick up the hydrogen and the electrons that have been created by the splitting of the water molecules, and they go over to the stroma where the CO2 is dissolved, and then they reduce that. But since the stroma environment does not exist, it does not happen. And you also require special enzymes in order to catalyze the reduction of the carbon dioxide molecule. So after all this, yeah, it's really good because he found out that carbon dioxide and water is processed separately. We have water, which is being processed in the granum. That's what he found out here. So he just had the granum only, just have the water and the electron acceptor. So the iron molecule, it compensates for the loss of the transport molecule A. So A and ADPH that was like replaced by iron or something, and that allows the electron to transfer. And after the electron transfer happens, only then can water be splitted, because if you have this split and you don't have the electrons to go anywhere else, then the reaction won't proceed as well. But if you have like this electron chain going on, oxygen will definitely be produced. So water, that's being split at the granum, and it involves the transfer of hydrogen ion and electrons to reduce either the iron ion, as seen here, or NADPH, not NADPH, NADP, if it's like in an actual plant that's alive. Next, carbon dioxide that is located within the stroma. That's obvious because, well, it's not involved in the granum reaction, so it has to have a role in the stroma. And yeah, water is the source of oxygen evolution, as you can see through this reaction here. So again, to recap everything, H plus an electron that gets taken up by the transport molecule. The transport molecule moves to the stroma where the CO2 is, and we have the transfer of H2 from the A transport molecule to the CO2, and that generates carbohydrates and sugars. However, this process is not as simple as it is shown here. This is the dark reaction, or the Calvin cycle, which is an even more complicated process than we see with this one line and one sentence here. Next, just some extra information before we finish off. Then there's a question of how did he manage to measure the production of oxygen, and that happens through the use of hemoglobin So his research was not only in plants, but he also focuses on hemoglobin. So he manages to take his plant research and hemoglobin research and fuse them into something that he could research with both at the time. So he used HB, hemoglobin, and he measured the saturation. So as you can see, as the dissolved oxygen starts to increase, that corresponds with an increase in saturation, as you can see from the graph here. So when hemoglobin is saturated, that's gonna demonstrate a different spectrogram. So you have IR spectroscopy or something like that. So you can tell whether oxygen has been absorbed by hemoglobin or oxygen has not been absorbed, and you can tell the specific percentage based on the spectroscopical results. And that's what he did. Because people before, they used bacteria. They used oxygen-sensitive bacteria. So I think that the bacteria would glow when you bubble oxygen through it. And that was used to indicate the presence of oxygen, but that didn't measure it quantitatively. It's just a yes or no. Was oxygen evolved? Yes or no. This was a quantitative measurement, which meant that you can actually tell how much oxygen was there. That's something that he pioneered as well. So this DCPIP molecule, which I think is dichlorophenol indophenol, it's a dye that changes color whether it is reduced or whether it is oxidized. It is something made by it's something made by the scientist himself, Robin Hill. It's made by him. And he developed it as a way to further study the photosynthetic reaction. And while it is oxidized, which is in this form here, it's blue in solution. When it is reduced, it turns colorless. So as you can see, the DCPIP dye added to a chlorophyll content sample, the blue turns to a green. That's not because this thing is green, this thing is colorless. The green comes from the chlorophyll that is within this solution here. So not because this is green, this is colorless. You should remember that. And DCPIP is now something that is used when in high school or in college, when you're asked to do the exact same reaction, you're most likely not going to do it with the iron like he did. You're not going to use the iron just because it's hard to determine whether the iron was reduced or oxidized. But now you have a spectroscopic method. You can immediately see the color change, and that way you know that the reaction is working just the way it was intended to do. So that's it. This experiment is really it really helped. It boosted a lot in terms of knowledge that we don't know yet. Before, we just have 5 sentences to summarize the process of photosynthesis, and now we have paragraphs that can explain the processes more, and that's all thanks to this one experiment. And it's a pretty simple one. You can even do it in your high school lab. So yeah, I think it's pretty interesting to discuss it, especially because it's so simple and yet it can provide lots of information. So yeah, you're gonna see a lot more experiments like this in the future. But for now, goodbye.